Hello, thank you for attending this virtual field day. I want to talk about some of my work with the evaluation of biological and conventional pesticides for Fusarium wilt management on tomato and watermelon. My name is Joseph Carrillo, and I'm currently a postdoc in Gary Villad's lab. So I want to introduce the pathogen. We're talking about Fusarium wilts in vegetable production. This is caused by Fusarium oxysporum. They have special forms, which are also called Forma specialis. Traditionally, they're named off which host they infect. Currently, there are over 58 known special forms. The first one I'll introduce to you today is on tomato. This is Fusarium oxysporum Forma specialis lycoprocessi. I'll refer to it as FOL. This is some of the damage it causes on tomato. You can see death that it has caused and some classic chlorosis and wilting here. It has three races, 1, 2, and 3. These races develop through the cultivation of tomato. Over time, we've developed resistance to this pathogen, and currently, most commercial varieties are going to have race 2 resistance, but the pathogen evolves to overcome that, and now a lot of population we have out there are race 3, which are causing some issues for tomato growing. Watermelon is a Fusarium oxysporum Forma specialis nivium. You can see some of the damage it causes here. It causes a lot of wilting, early plant death in watermelon. Nivium comes from the Latin word, it means snow, which you typically see on watermelon crowns during some infections. In certain conditions, you'll see the mycelium kind of coming out of the crown, which looks white, kind of like snow. I'm going to call this fawn from here on out. And the races here, there are 4 races, 0, 1, 2, and 3. Again, they arise as a result of our cultivation because we're building resistance against them. Signs and symptoms on tomato. The infection is going to occur through propagules that are in the soil, and they infect the rootlets. The optimal temperature is 82 Fahrenheit. Generally, this is going to be a pathogen that prefers a little bit warmer temperatures. Once it's in the roots, it's going to invade xylem tissue and invade the water transport. It can cause stenting, but the most classic symptom you're going to see is chlorosis. This is going to affect typically one side of the plant at first, as shown here, and also it can affect upper parts, as seen here. But typically it's going to be on one side first more than the other. What you can look for on leaves is this classic asymmetrical chlorosis, which you get this golden color, which you've seen in the previous pictures too, but you'll see it on one side and not the other. This can happen on single leaves, the whole leaflet here, but it'll be asymmetrical as opposed to like nutrient deficiency symptoms, which will be typically symmetrical. And you'll get some brown internal discoloration in this greenhouse experiment I did. This is the non-inoculated control. You can see no browning here, but this is inoculated with full. You see some browning in the tissue, and then on the cross section you can see some of the browning there. Symptoms are usually going to appear later in the season, and they can cause some early ripening of fruit and plant death. On watermelon, we get a similar infection through the soil into the roots. The optimal is going to be 77 to 81 degrees Fahrenheit, which is a little bit cooler than full. Fawn is going to prepare a little bit cooler temperatures. Also invades the xylem, or the water transport system, and these are some of the symptoms you'll see. Some early wilting, which can lead to plant death. You can also have some stunting without wilting, but this picture here kind of has a nice comparison of a healthy individual compared to early on wilted individual. You'll also get some gray in the foliage, chlorosis, and wilting like I mentioned. And of course, just like with FOL and any other Fusarium oxysporum, that's a pathogen, we're going to get some brown internal discoloration as they invade the xylem tissue. Symptoms usually going to appear 3 to 4 weeks and can cause early death before fruit set. Diagnosing Fusarium wilts typically is going to be diagnosed in the field through scouting using visual symptoms and signs. In the lab, the plant samples and soils can be isolated on media, which we can purify to get pure cultures, just like these two shown here. Typically on culture media that has a base of dextrose, Fusarium oxysporum is going to exhibit this kind of purple color, which you can identify it as Fusarium oxysporum. However, molecular identification using DNA has become more useful to us because we can distinguish pathogen versus non-pathogen. There's actually Fusarium oxysporum out there that exists in the soil that aren't pathogens, but this identifying the DNA, which the pathogens have that the non-pathogens don't, allow us to determine if the sample is a pathogen or not. So current management strategies are going to be cultural management, using resistant cultivars, clean transplants, grafting, which can be done in tomato and watermelon, using a scion of your desired fruit quality. The rootstock will have the resistance to the disease. Soil pH, if you keep it on the high side, 6.5 to 7, that's going to reduce your incidence. The Fusarium molds typically prefer more acidic soil. And crop rotation is difficult because of these resting structures that they have called chlamydospores. They can survive up to 10 years in the soil, which makes crop rotation difficult. Chemical management is done through soil fumigation using nematicides. They are also effective on foal and other Fusarium molds. Piclor 60 is a common one used, but there are other fumigants available in the Florida Grower Handbook. Chemical fungicides are used in watermelon that are applied through drench or drip, plus a follow-up foliar application. And I'll be talking about that a little bit later. And also biological control is another management strategy. This is the use of organisms to control other organisms. I'll be talking about this a little bit later and how we're trying to approach that. So first, I'm going to talk about a field trial that we conducted in the fall of 2009 and spring of 2020. The watermelon variety Jade Star was inoculated with fawn. Application using 6 treatments. 5 reps was done in the fall and repeated in the spring. The treatments selected were Proline, which is the industry standard, and this was applied through a drip with a follow-up foliar application. And we were experimenting with a new product, Miravis Prime, which we used some low concentrations and high concentrations, which are listed here. We did some, with a low and high concentration, we did drip at planting and then foliar 7 days after. With drench at planting, we did, or with the high concentration, we did drench at planting and then foliar 7 days after. And we also did a drench at planting and then drip 7 days after. So again, this is an experimental treatment with Miravis Prime to see how effective it can be in controlling this Fusarium on watermelon. So here, this figure shows the disease progress over time, which is also known as the area under disease progress curve. The comparisons between the groups indicate that Proline and Miravis Prime performed similarly, and they both significantly reduced disease compared to the control here, which is marked with a different letter. So we didn't see any differences in the application methods, but we did see similar effective control of the pathogen as far as reducing the incidence compared to the control. Unfortunately, in this season, we weren't able to harvest because of the wildlife were eating our watermelon. So. But we did repeat in the spring, and we saw some more separations of these different methods of application. In particular, we saw the drench followed up by the foliar application had a significant reduction of disease compared to all other treatment groups. We also saw Proline performed, again, significantly less than control, but significantly better than the Miravis Prime treatments, which were done similarly at the high and low concentration using a drip followed up by a foliar application, as we did with Proline. While the drench applications with Miravis Prime both were more effective than Proline in the spring. And here's some images from the field trials. You can see the inoculated control. We have some plants that succumb to the disease, and overall a little bit less foliage than compared to the Miravis Prime shown here, and also the Proline, which did really well in this trial as well. So we were able to harvest at 7 weeks. We did a little bit earlier than normally, just to combat the wildlife issue we were having before, so we can actually make some comparisons of what we're recovering. We converted these values to tons per acre based on the plot size. Yield was significantly higher in both Miravis Prime treatments that included an initial drench, although the follow-up treatment was different. In this one, it was a follow-up foliar treatment. This one was a follow-up drip, but they performed better than the other groups, definitely better than the yield we got in the control. So conclusions and outlook. Miravis Prime is comparable to Proline in managing Fusarium wilt of watermelon, and the average tons per acre increased in Miravis Prime treatments that were initially drenched compared to the Proline treatments. And Miravis Prime offers different frac groups, which are 7 and 12 compared to Proline, which is only 7. And for the future, we're going to evaluate this product since it seems to be effective against fawn. We're going to evaluate it against full on tomato and see how well it does there. So now moving on to biological control of Fusarium wilt on tomato. There are many microorganisms such as fungi, bacteria, and viruses that are present in the soil. However, not all are bad. Some have qualities that can inhibit or outcompete plant pathogens, in some cases promote plant growth as well. And many products are commercially available. A lot of the products that are available will be either a bacteria, just like in this case, that produces some metabolites that are going to inhibit fungal growth, shown in this image. You can also have bacteria that are going to inhibit other bacteria from growing. And you can also have fungal products, in this case this is a trichoderma example, that can parasitize other fungi or potentially outcompete them or produce metabolites that can reduce bacterial growth and also reduce fungal growth. So there are options for biologicals against certain pathogens. So we're focused on trichoderma because there are a lot of products available, and specifically because they're labeled for control of Fusarium species. They do have a lot of different modes of actions, ranging from competition, where they can outcompete the pathogen in the environment, antibiosis, where they can produce something that's going to inhibit the pathogen growth. They can be endophytes, which can promote growth. They can induce resistance in the plant, which can help it resist pathogen infection. And also what we were focused on is mycoparasitism, looking at certain isolates that can colonize or parasitize other fungi. So we're looking for products or isolates that we can recover that are going to attack full and hopefully attack that resting structure is the goal. So biological control, some of the pros: minimize safety concerns. You don't have to worry as much about compared to chemical applications. It's environmentally friendly and economically sustainable. And also, they're OMRI listed products, which can be applied in organic settings. Some of the cons is they vary in cost. They may not establish in different environments, such as environments in Florida. And none of the ones that we've tested so far on full have worked as far as commercial products. So this ventured us to look into local biological control agents, and we're focused on trichoderma in particularly, because they can maybe be more established and better suited to survive in this particular environment here. And they may have similar qualities that are found in some of these commercial products, just they're better adapted to survive here. So we're looking into local agents for that. And this is just a picture of one of the example ones that we recovered. So some of the ones that we recovered we recovered them actually from local tomato fields. We recovered candidates narrowed down to 10 different species, and we applied them to this variety of tomato in a transplant stage. We also compared this to local strains in the commercial isolate T22 as inoculated control. And 2 weeks later, we inoculated full race 3 into the transplants after they're colonized with trichoderma first. And at the same time, before the plants went in, we applied the same trichoderma to the plots 2 weeks before planting, like a pre-plant drip application. Then we followed up with drenches at 0, 2, and 4 weeks to try to ensure that the trichoderma species of interest was in the soil. And we evaluated disease incidence, severity, and yield. So here we can see disease over time was significantly reduced by one isolate in particular, Trichoderma longibrachiatum, which was significantly reduced disease incidence compared to all treatments. And here you can see the non-treated control, which had a higher incidence over time, and then the T22 commercial strain actually had higher incidence than the control. But we're excited about and are going to repeat is looking at this particular isolate Trichoderma longibrachiatum. This is some images 11 weeks post-planting, the inoculated control, which you can see some yellowing and some dieback here compared to longibrachiatum. Looks a little in better shape. And this is another view here. We can see in the inoculated control, we had a couple plants succumb here. We have some yellowing, and then we have Longibrachiatum in the back here. Although it did get some infection, it overall produced better, or grew better. So conclusions and outlook. Trichoderma longibrachiatum is a potential candidate to manage Fusarium wilt. It performs significantly better than the commercial strain in control in reducing disease incidence. Unfortunately, we didn't see any significant differences in yield this time around off of 2 harvests, but we will repeat this in the spring season to see if there's any differences in the different environmental situations in the different seasons. And Trichoderma longibrachiatum actually, from previous research, prefers warmer temperatures, so it may be ideal for Florida for us to develop this as a biological control targeted in this region. But we still need to do some more work on this one. So I thank you for your attention, and I look forward to answering your questions in the live Q&A. Thank you.